Pause the video if you need to. We'll give you 15 seconds to read the question, and then we'll go through the explanation. All right, so we have a researcher that's studying the relationship between pattern recognition receptors present within host cells and Crohn's disease. The pattern recognition receptor nucleotide binding oligomerization domain protein 2, NOD2, has been shown to detect conserved motifs in bacterial peptidoglycans, such as intracellular muramyl dipeptide, and promote activation of host inflammatory pathways. So let me stop right there, because this is a situation where you want to stick to the things you know, because you're going to see things that are kind of thrown at you in questions that, are different than anything you've seen in your study material. So, you want to stick to the things you know. So what do we know? We know a little bit about pattern recognition receptors, which I'll talk about in a minute. Okay. And you might have heard of NOD2. If you haven't, that's okay. We'll talk about that. The bottom line is, they're basically saying here that the pattern recognition receptors, one example of the pattern recognition receptors is NOD2, right? So we can kind of write that down. So pattern recognition receptors, and we'll talk about these in a second, they're basically telling us that an example of a pattern recognition receptor is NOD2, okay? And NOD2 is going to detect conserved motifs in bacterial peptidoglycans, and they give you an example here, and promote activation of host pro-inflammatory pathways. Even if you didn't read that, right, you should know, right, from the video series we've kind of just covered in this chapter, what pattern recognition receptors are and what they do, right? So pattern recognition receptors are going to recognize pathogen-associated molecular patterns, or PAMPs, right? They can also recognize damage-associated molecular patterns, right? So these receptors can kind of recognize these very non-specific patterns in certain bacteria or viruses, for example, right? And we kind of talked about some of these. So we said, look, there's some PAMPs, for example, LPS, right? And gram-negative bacteria. That's very non-specific. A lot of different gram-negative bacteria, right? They're going to have LPS, and we can recognize LPS non-specifically. It's not a specific antigen and a specific idiotype that are coming together. This is just a, LPS in general sets off this host inflammatory cascade. It's very non-specific, right? So LPS can do this. We said viral nucleic acids can do this. Even bacterial flagellin is another classic example. And then the damage-associated molecular patterns are usually things from host cells. So, if a host cell is, has defective DNA repair or something along those lines, for example, there's going to be very non-specific patterns that can be recognized to target that host cell for destruction. Again, these are non-specific. So if something is non-specific, it's generally going to be part of the innate immune system, right? So this whole process is really part of the innate immune system, because again, these are non-specific patterns that kind of set off that initial cascade. When you have a pattern recognition receptor like NOD2 or a toll-like receptor, which we kind of talked about, TLR, toll-like receptor, these can recognize these non-specific patterns. And what they end up doing is, once these bind together, right, once LPS binds to this toll-like receptor, or flagellin binds to, something like NOD2, whatever it is, what ends up happening here is we eventually end up getting an inflammatory cascade. Okay, and this is kind of what we talked about in the last video. And the way this all starts is by increasing transcription of NF-κB, which is particularly high yield. Okay, we talked about this being a very important step in starting this inflammatory cascade. And then this kind of sets off multiple cytokines like interleukin-1 to change temperature set point, interleukin-6, right, TNF-alpha, all of these kind of acute phase inflammatory reactants. Okay, and so this is kind of how this whole process works, and that's what you're kind of expected to know. You're expected to know this is very non-specific, sets off an inflammatory cascade through the regulation of NF-κB, and then kind of the big ones to remember here are TNF-alpha, interleukin-6, interleukin-1, maybe even some interleukin-8, right, for chemotaxis. With all that being said, going back into the question here, we're going to read this line again. The pattern recognition receptor, NOD2, has been shown to detect conserved motifs in bacterial peptidoglycans, where you just said, we already know that, right? Conserved motifs, right? Things like flagellin, LPS, etc., right? That's something we already know. And that gives you an example of one here, intracellular muramyl dipeptide. And when doing this, it promotes activation of host pro-inflammatory pathways. Okay, that's very important. So NOD2 has been known to be highly expressed in the Paneth cells of the intestine. That might have something to do with the fact that they're looking at this relationship in Crohn's disease, right? The researcher attempts to induce mutations to further elucidate the effects of ileal inflammation on NOD2 deficient mice. So we want to see the effects of inflammation on NOD2 deficient mice. Key word here is deficient, right? So these mice do not have this presumably this gene that codes for this NOD2 receptor, right? So based on the information above, which of the following would most likely be impaired in NOD2 deficient mice? Okay, what would most likely be impaired? Now, if you kind of understand this, which is what we kind of talked about, it should make sense that NF-κB activity would actually be impaired. And you could say, well, why is that? Well, remember, these mice don't have this NOD2 receptor, right? So if they don't have this NOD2 receptor, we can make the assumption that, look, if I don't have this receptor, if I'm deficient in it or it's dysfunctioning, whatever it is, I'm going to have an issue setting off an inflammatory cascade, right? I'm going to have an issue setting off an inflammatory cascade. So what will happen is potentially, right, I'm not able to go through this next step to upregulate NF-κB because I don't have the receptor to make that whole process happen. I don't have the receptor that then binds to, in this case, this intracellular muramyl dipeptide, right, to set off this inflammatory cascade. I don't have the receptor to bind to flagellin or viral nucleic acids or LPS to kind of go through these steps, right? So if I don't have that receptor, then everything downstream is affected. So I might have, low levels of interleukin-1 in the setting of an infection, or interleukin-6 or TNF-alpha. In this case, it has to do more with Crohn's disease, but the point is still the same. The point is that we can't set off the inflammatory cascade, and the point is that you should understand that these pattern recognition receptors are associated with the innate immune system and setting off the inflammatory cascade through the regulation of NF-κB. Now let's quickly just go through some of these other options here. And if you're just kind of watching these videos sequentially, we haven't really talked about some of these other things yet, but I'll just give you a little bit of a rundown as to why they're not correct. So B7 protein binding to CD28. So B7 and CD28 are kind of linked because these two come together essentially to perform a co-stimulatory signal. And this has to do with the activation of T cells, which ends up being part of the adaptive immune system. And this is done in situations where you have a specific antigen, right? And you might be creating a memory response, for example. In these situations, though, again, this is going to be more adaptive immune system, not really directly related to the NOD2 receptor or this innate immune pathway that we're talking about. Okay, so not directly related to this and would be unlikely to be directly affected. Certainly not the best choice here. CD40 membrane receptor activity. Now, this is actually a pretty big one. So CD40 and CD40 ligand, which we're going to talk a lot about in this series, kind of come together to allow for class switching of antibodies. Now, remember what we kind of talked about? We said, look, IgM is kind of like this generic antibody that we have. And we can make more specific forms of our antibodies by adjusting the isotype. Okay, so we can adjust the isotype of the antibody to make things like IgG, right? Great for our memory responses, IgA at mucosal surfaces, IgE, right, etc. So we can make these different types of antibodies, but to do this, to undergo this process, we have to undergo class switching. Okay, so we have to do some kind of class switching to let this happen, to change the isotype. And to do that, the key receptor binding to do that is going to be the CD40 ligand, which is going to kind of set off this cascade. And I'll go through the entire pathway, all the details, as we kind of progress through this unit. But what I just want you to understand here too, since we're on the topic of this, I don't want to go too far down into the details yet. So you have a heavy chain, which I'm drawing in black here, and then you also have a light chain. Okay, so you have a heavy chain, and then you have a light chain. And the heavy chain has kind of two regions here. So this pink region is the constant region. Okay, so we just say this is the FC region, the constant region. And then we also have a FAB region, an FAB region. So the light chain and this part of the heavy chain are going to make up the FAB region or the FAB region. When you're changing the isotype, right, this is what we're doing to go from like IgM to IgG, right? When you're changing the isotype, you're going to be changing the FC region here, okay, when you're changing the isotype. Now, if you changed the idiotype or changed the variable region where this antibody can bind, that's going to be in this area, in this FAB region, okay? So this FAB region is going to contain, hypervariable regions that bind to very specific antigens that underwent VDJ recombination, whereas the FC portion is going to be more regulated for complement binding and the isotype. Anyway, the point of this is because IgM is kind of like our generic antibody, IgM can be thought of as part of the innate immune system, right? Because it's kind of our generic antibody. But for us to go to class switching, that's where we're now kind of transitioning into the adaptive immune system, right? Because we're creating antibodies for memory responses, making antibodies like IgG, for example. And so we can say that IgG is actually more part of the adaptive immune response because we require class switching. And again, we're primarily talking in this question about the innate immune system specifically, right, with the NOD2 receptor. The class switching would be kind of out of the scope of what's being tested, in this study. All right, so if we look at option D, this is really just along the same lines of option C. It sounds kind of complicated, alterations within the heavy chain constant domain of immunoglobulins. What does that mean? It's just what we just said, right? Here's the heavy chain, right? This is the heavy chain, this black line. Here's the constant domain. If I change the constant domain, I end up with IgG, IgA, IgE, right? Some different form of the immunoglobulin. So this would not be the correct answer also for the same reasons, because it would have to do with class switching, adaptive immune response. Now the last option on here, terminal deoxynucleotidal transferase activity. Now what this is, and you do have to know a little bit about this, but you don't have to know a ton of details, but what this is it's a DNA polymerase that kind of randomly adds nucleotides to immature T and B cells. Now the thing about terminal deoxynucleotidal transferase, number one, you sometimes kind of think about this when you're thinking about things like RAG1 and the RAG2 genes, which are basically like recombinase genes that allow for VDJ recombination to create this kind of hypervariable region here to make all different forms of these, idiotypes, for example. Now, this process, is not directly related in any way to what we're talking about with the pattern recognition receptor, so it's clearly not the best answer. But the other thing I want you to understand when you see TDT, right? If you see TDT positive, in a question stem or a lab value, you start to think about, a little bit about, especially in the setting of blastic tissue, you start to think about acute lymphoblastic leukemia. And one of the ways that we start to differentiate the different forms of leukemia is you can look at acute myelogenous leukemia. Typically, AML is TDT negative. And that's because the blastic cells in AML typically are from the myeloid lineage, not the lymphoid lineage, right? The lymphoid lineage would typically have more cells that might have this TDT, this polymerase, essentially, that's allowing for VDJ recombination in these very immature blastic cells, right? Blastic cells are immature. So the point is that you can see TDT positivity in ALL, especially in the setting of blastic tissue. That's what I want you to remember. But again, this is not directly related or linked to this question, right? If you understand this pathway that we talked about in the previous videos in this chapter, you can pretty much solve these questions where they start to throw some information at you that seems a little convoluted, seems a little complicated. Just stick to what you know. Look for the things that you know and stick to the pathways that you've kind of gone through already, and usually you can decipher some of this.